We continue the migratory path of sperm by asking on-campus students to tell us what they thought about fertilization. The next video shows us what they had to say.
Fertilization is when a sperm cell meets the oocyte and burrows down through the layers and it has the fusion and then creates the fertilized egg.
When the sperm meets the egg?
Sperm meets the egg.
They go inside it. So instead of dying and their tails drop off like tadpoles, they make it inside, I think.
No.
When the sperm meets the egg, it gets fertilized.
Formation of an embryo and a zygote, from a zygote.
Yeah.
Formation of a zygote, yeah.
After their incredible journey through the male and female reproductive tracts, sperm are now approaching their ultimate destination. And this is the secondary oocyte, or the egg, waiting in the expanded ampulla of the oviduct, also referred to as the uterine tube or fallopian tube. Of the millions that began the journey, only 100 or so sperm will arrive safely and compete for their chance at fertilization. As sperm near the end of their journey in the oviduct, their focus changes from just finding the egg to preparing for the ultimate event, fertilization. The preparations that sperm undergo are known as sperm activation or capacitation. These are the acquisition of the ability of a sperm to recognise and interact with the egg. The exact triggers for sperm activation are not fully understood, but they're known to involve environmental factors in the female reproductive tract. During sperm activation, or capacitation, the membranes over the sperm head become less stable and more fluid, which subsequently enables the release of enzymes during the acrosome reaction, and the sperm tails become capable of more frantic and forceful movements known as hypermotility. Once activated, sperm are equipped and ready for their meeting with the egg, but first they must navigate their way through the layers immediately surrounding and protecting it. The final step in the sperm's quest to meet the oocyte involve firstly penetrating the cumulus to reach the zona pellucida surrounding the oocyte, then binding to the zona, where membrane proteins in the sperm head bind to ZP3 receptors of the zona pellucida through a key and lock mechanism, which brings about the third stage, which is the acrosome reaction. This is where sperm membranes vesiculate. This means that pores form in the membrane, and enzymes are released from the sperm head to facilitate the fourth stage, which is zona pellucida penetration. Here, a combination of enzymatic digestion and thrusting of the sperm tail enables the sperm to pass through the zona pellucida to enter the perivitelline space, immediately surrounding the oocyte. Finally, the stage is set for fertilization. Fertilization begins with the physical contact between a sperm and the oocyte. This contact involves a region of the sperm head from which the plasma membrane has been removed in the acrosome reaction, binding to the oocyte plasma membrane in an event known as sperm-oocyte binding. Binding then triggers additional events involving the oocyte that prevent the participation of additional sperm in fertilization, which is called polyspermy, and completes the fertilization process. The stages of fertilization are as follows. Firstly, the sperm are incorporated into the oocyte cytoplasm, or commonly called the ooplasm. Both the sperm head and tail are taken into the ooplasm. Secondly, after a single sperm has entered the oocyte, the egg depolarizes. Depolarization means that there is an increase in the level of free intracellular calcium within the oocyte. This causes a barrier to prevent multiple sperm entering the egg, and this is very important because polyspermy, as I've already mentioned earlier, would lead to a condition known as polyploidy, in which the zygote would have too much genetic information and is often not viable. The third stage is when you get reactivation of meiosis in the secondary oocyte and extrusion of the second polar body. In the fourth stage, decondensation of the sperm and oocyte nuclei form the male and female pronuclei. The fifth stage is syngamy, and this is where you get merging of the pronuclei and mixing of the male and female chromosomes. Fertilization is now complete. The fertilized egg is a diploid zygote in readiness for the first mitotic cell division. The term implantation is used to describe the process of attachment of the fertilized egg to the wall of the uterus at the start of pregnancy. In humans, implantation of a fertilized ovum is most likely to occur around 9 days after ovulation. However, this can range to between 6 and 12 days. The zygote that is formed following fertilization remains within the zona pellucida and begins to move along the oviduct to the uterine cavity. A lot of significant changes take place during this journey, which will be outlined in this video. First, cleavage division occurs during day 1 following fertilization, and the 2-cell stage with 2 identical blastomeres is formed. Blastomeres are the cells that are produced during cleavage of a zygote and that form the morula, which is a solid ball of identical cells. Following this, there is further division to 4 identical blastomeres. After 3 days, the blastomeres undergo compaction. There is no increase in the overall size of the conceptus during this time. Rather, the cells become smaller. The morula typically reaches the uterus on day 4 and the cells have continued to divide. Over the next few days, these identical cells divide further, forming a blastocyst, which is a hollow ball with an inner cavity containing nutritious fluid called the blastocele. It is important to point out that the wall of the uterus contains glands that secrete nutritious fluid, which initiates the formation of a blastocyst. Some cells are displaced centrally from the periphery of the blastocyst and become the inner cell mass. Cells are no longer identical in size and shape, with 2 different populations now present. The next stage is called hatching, and by the end of the 5th day, the blastocyst is now ready to implant into the uterus. Its protective zona pellucida thins and ruptures, and blastocyst hatches. The blastocyst now has an outer layer of cells called the trophoblast, which are responsible for providing nutrients to the developing embryo. The inner cell mass is still present and is protected from the outside world by the trophoblast cells, and in time will go on to form the embryo. The trophoblast cells give rise to the chorion, which is one of the membranes that surround the fetus while it is still being formed, and also give rise to the placenta that nourishes and maintains the fetus through the umbilical cord. As the blastocyst makes its journey through the oviduct, it provokes a response of considerable magnitude in the uterus. The endometrium of the uterus changes shape and produces a hospitable environment for the embryo and becomes receptive to implantation. The endometrium swells, its glands secrete nutrients, and more blood vessels develop, providing a hospitable environment for the developing embryo. There is only a short window of opportunity that the uterine lining welcomes the developing blastocyst, which is usually between 6 to 10 days after ovulation. Apposition is the very first loose connection between the blastocyst and the endometrium. After the apposition of the free blastocyst at the uterine epithelial surface, the microvilli on the surface of the outermost trophoblast cells interact with the lining of the uterus. This is called adhesion, and the blastocyst realigns itself so that the inner cell mass is adjacent to the uterine wall and the trophoblast over the inner cell mass contacts the endometrium. This orientation of the blastocyst is very important because the inner cell mass needs to be very close to the blood vessels and nutrient supply within the endometrium, as it has high metabolic demands and is part of the blastocyst that will eventually go on to develop into the embryo. Adhesion generally occurs on or around the 7th day after fertilization and results in the blastocyst being firmly fixed to the uterine epithelium. Following this attachment, the cells of the trophoblast begin to release digestive enzymes that dig a hole inside the endometrium to enable access for the developing embryo. These trophoblast cells start invading between the endometrial cells, which initially attaches the blastocyst to the uterine surface and then gradually buries itself deeper into the uterine wall. This is called invasion, and during this time, more changes take place to the cellular trophoblast. The inner cell mass separates from the trophoblast, creating a richly nutritious fluid-filled chamber called the amniotic cavity. Once inside the implantation hole, the endometrium is then sealed off. By about day 10, the entire developing embryo is completely inside the endometrium.